Just imagine if you can find drug targets by screening thousands or hundred thousand molecules, million molecules against a particular disease target, against a particular receptor, without even having a wet lab, without doing any kind of wet lab experiments. You can do that by just using your laptop. In modern drug discovery system, the screening of these molecules inside the lab is highly difficult. It's highly challenging because cost is involved, various models that are required. You need money to actually grow these model systems, including, let's say, cells, including mammalian cells, including animals, if you want to test those compounds. But here is the deal. By using virtual screening methodology, you can actually test these compounds inside a computer system. That is exactly where the virtual screening methodology becomes a game-changing system. It will help researchers identifying lead molecules from library of millions of compounds. And everything can be done by using a simple laptop, by using a simple system that you have right now. So I welcome you to Virtual Screening for Drug Discovery course at ResearchGoal. It is not a theoretical course. It will provide you hands-on practical training, and you will be using real data sets. You will be downloading those data sets. You will be filtering out compounds. You will do much more and identify target compounds against the target protein that you have. And in this course, you will learn step-by-steps the things that will help you to do virtual screening inside the system that you have. We will start with using Linux system inside the Windows system. I'll show you how you can use Linux environment inside the Windows system. And let me tell you, this is very, very easy, and there is no need for you to actually have a different system for this particular course. You can work with the Windows system that you have and then apply Linux environment inside your Windows system. After that, we will be working with basic Linux commands. I'll be showing you that what are the basic Linux commands that you'll be using, and I'll give you detailed overview of all those commands and how you can use them inside the project. Next thing we'll do, we'll get those compounds, different compounds from different databases like ChemBL. So we'll have those filtered compounds that we are going to use in our virtual screening platform. The next thing that I'm going to show you is how you can handle and convert different file formats like SDF, PDB, or PDB-QT. You will also be automating different tasks using scripts. Finally, we will perform large-scale virtual screening pipeline. And everything we'll do in our laptop only. So with that particular experiment, computational experiments, we will have the lead compounds against the target receptor that we are using. As I've told you earlier, this is not a theoretical course. It will give you hands-on practical experience, and you will work like a real drug discovery scientist, and you will get actual lead compounds that probably are the lead compounds for experimental validations against your target receptor. Now, coming on to the prerequisite, because there are some prerequisites for this course, you have to complete the course that is molecular docking for drug discovery. Because in this one, we are using more like a command-based pipeline to actually do work without understanding how the protein structure is there, how we can perform the grid box, how we can get other files that are required for automating all those things. So my humble request is, please complete the course that is on molecular docking that I've already created. If you have performed molecular docking by yourself using Autodoc Vina, you can directly jump onto this particular course. There is no additional thing that you'll be needing. But if you want to learn everything, maybe 100% of this particular course, then please have that course done, which is molecular docking for drug discovery. I made that detailed course that is available on ResearchGoal, and then complete this particular course. So in that course, what we have taught is we have shown you how you can actually prepare ligand files, one ligand file, how you can prepare one receptor file. In this course, we will be showing you how you can perform large-scale molecular docking, which is also known as virtual screening. So that is why that course is really, really essential to complete this particular course. Another important aspect of this particular course is you will be automating things. You will not be doing manual, copy pasting, manual creating folders and all. You will be working in a command line like a professional, like an industry grade professional, where you will be automating tasks. You will be, writing scripts that will help you create the automated workflow for your project. So it will become very, very handy for you to actually do virtual screening, large scale virtual screening even with your laptop. If you have a bigger system, that will be a plus advantage, but everything will remain the same for all kind of virtual screening projects. This course is designed for undergraduate students, postgraduate students, PhD scholars, early researchers who want to know virtual screening. Everyone can actually do all these experiments. What they need is just a laptop. If you have Windows laptop, you are good to go. You have a Linux-based system, you're good to go. Nothing else is required. Everything I'm going to show you how you can download these softwares, how you can run the project from scratch. So basically, anyone who is interested in AI-driven computational drug discovery, this course is for you. By the end of this particular course, you will be able to design the workflow for your own project, virtual screening project. You should be able to download the ligand files that you need. You should be able to automate or write scripts that will help you refine your ligand files or convert those ligand files in the format that is required for virtual screening. You should be able to analyze and filter out the results, and you should be able to identify the molecules and understand those molecules in detail, because we'll also show you how you can do analysis of your virtual screening results, and that is really powerful. And let me tell you again, if you're really serious about computational drug discovery, if you are interested in designing your own workflow and learning more about AI-based computational drug discovery, this course is for you. And as you already know, we don't have very high prices for our courses, but we have really good quality of our courses. So please join the course, and do let me know in the comment section if you are facing any kind of issues. I'll be helping you out with any issues you are facing. If you need mentorship, we can have one-on-one session. I'll help you designing your own research project, and I'm looking forward to meet you in that particular course. Again, thank you so much for supporting Research Goal. We'll see you in the course.